Start this end of the blue line. Which is it, 5 or 3? It's a 5. That's correct. And that makes the odd end of the blue line 3. That's right. So and how do you know that? 5 prime to 3 prime. So Barbara, thank you. How do you know that? Because the rule says you polymerize 5 prime to 3 prime. That is the law. Thou shalt always make thine RNA 5 prime to 3 prime. Whatever direction it points, you start at a 5 and go to a 3. All right. So let me do the next question, which is, so to hand out some more points. The next question is, okay, so which DNA strand are we pairing with, the top or the bottom? So Jairus, are you here? Yes. So tell me, are we pairing with the top or the bottom? We're pairing with the bottom strand. Why? Because that one goes 3 prime, 5 prime another way. So 5 to 3 on the one way. The other part of the law says, thou shalt always make thine base pairs in anti-parallel strands. So you can only pair with the bottom. All right, that's also why you can't pair with the top. If you tried to pair with the top, they go in 5 to 3 the same direction. That's a big no-no. Right? So you have to have them paired like this. Questions about that? All right, that's the secret of this. That's why you only get one RNA. All right, last question. Since we're paired, so we're going to be pairing with the bottom strand, all right? Let's just do one last thing. Victoria, what's the first nucleotide going to be? Well, Victoria, are you here? Victoria, hi. What's that first nucleotide going to be? The one that goes here. Pardon? I got a T. That's almost right. But because if it was DNA, it would be a T. What's the trick with RNA? It's a U. It was sort of almost a trick question. I will thank you for that. It is, in fact, a U. And this next base is A, C, G, C. All right. Cool. So questions about that, because that's super important. You only get one. Actually, let me draw this really clearly. Those are the little base pairs. Right? You can only make one RNA. One strand is ignored. That's just the way it is. It seems uneconomical, but that's life. It's just the way it is. Right? Questions? Yeah. Is it always, very good question. Levi says, is it always going to be the bottom strand you pair with? No, it depends on your 5s and 3s. If I had the promoter pointing this way, you'd pair with the top strand. If I swapped my 5s and 3s and went this way, it would be the top strand also. So it depends on your 5s and 3s. Exactly. All right. Yeah. So are there any genes that have promoters going the other way? In practice, no. As when we learn about translation, it's very difficult to make a DNA sequence that works read both ways, and it gives a reasonable protein. All right. Hey, shh, everybody. Let me add a couple details to this. Let me do this in black. This thing is called the coding strand. It's ignored by, hey, if I can hear you guys, then it means it's really loud. Thanks. This is ignored by RNA polymerase. And this thing is called the transcribed strand because it's read by RNA Paul. Okay? There is a nifty little trick that the mRNA has the same 5 prime, 3 prime sequence as the coding strand, except the T's are replaced by U's. All right? That's a neat little trick. When you get good at this and trying to figure out what protein a gene will make, you can use that trick. You just read the coding strand and mentally read it as UACGC. It's the same as the RNA. There's a couple more things I want to say, and then We'll wrap it up. So number 4, RNA Paul continues to the terminator. Number 5, mRNA is released from RNA polymerase. And as a note, only one strand is made. I don't know who it is. I keep asking people to be quiet. I can really hear you nice and loud and clear. So thanks. Now it's gotten quiet. Only one strand is made, and it usually encodes one protein only, because there's always exceptions in biology. There's a couple notes I want to make which raise the things I said at the very beginning of class, that not all DNA is transcribed. It turns out some of that ever. That is, there are, let me write that as a better R, there are spaces between genes, promoters, terminators, etc. There's actually a lot of stuff in your genome that is never, ever transcribed. And not all your DNA is transcribed all the time. That is, genes are turned on or off by controlling transcription. This is called gene regulation. And I did my PhD thesis on part of this. It turns out gene regulation is a huge subject in biology. And that is basically, if there's no mRNA made, that means that no protein for that gene is made. If, on the other hand, the mRNA is made, the result is the protein is made, sort of maybe. Depends. And so how does a cell know which genes to transcribe? It turns out there are proteins that bind to promoters that turn those genes on and off. Well, so then how does it know to transcribe those proteins? Well, other proteins are transcribed or not. In the end, it turns out there are various factors that control whether genes are expressed or not. And that understanding how the control of gene expression is a huge part of biological research, because it's understanding why different cells do different things and why they do the right things and sometimes the wrong things. All right, we are out of time, so let me do the clicker question, and we will call it a day.